welcome everyone who comes into our room here. We are welcoming everyone to the monthly West Coast Metabolomics Center Invited Speaker Seminar. My name is Oliver Fien. I'm director of the center, and for those who are here the first time, please use the chat room. For when the speaker speaks, you can already make questions, post questions into the chat room. And Dr. Arpana Vania, who you see here already on the screen or in the picture, she will then present and guide you through those questions. So please, please, please do not forget to post questions. Otherwise, the speaker will think we were not listening. And in terms of our speaker today, so he has already done quite a bit since his PhD. So he did his PhD at the same unit. So that's the Analytical Biogeochemistry at the Helmholtz Center in Munich, which is the German Research Center on Environmental Health from 2009 to 2012. And then he did a postdoctoral fellow at the same unit. And in 2015 became a staff scientist at the same unit, responsible for UPLC method development and instrumentation, C. elegans cultivation. And then he started training and supervising guests and collaborating partners. Since then, since 2015, he is the chair of, or he's at the chair of analytical food chemistry at the Technical University in Munich for the School of Life Sciences. He's doing research on methods for metabolomic analysis from limited sample amounts, data and analytical strategies for metabolite ID and data interpretation, supervising bachelor, master, and PhD students, and teaching at the University of Munich. And his current position is he is the deputy head of metabolomics and the proteomics core and executive manager for metabolomics at the Helmholtz Center in Munich. So he stayed there all his scientific career, which is kind of cool. In Bavaria, German. So I'm not sure if he wears lederhosen. Do you do?
Sorry? I am not wearing my lederhosen today. No, unfortunately not. So if you would have asked for it, I would wear it.
But you do own a pair?
Not only one pair, several pairs. As
okay.
A Bavarian-born, Bavarian-living person, you have several pairs of them.
Good for you. Good for you. So welcome, Bavaria. Welcome, Dr. Michael Witting. The floor is yours.
Thank you very much, Oliver. Thank you very much for this kind introduction. So let's see if I can share my screen. It worked before, and it seems to work now.
Yes, right? We see you. Mm-hmm.
Perfect. So thanks for the kind introduction. Yeah, and you're totally right. I'm what we call in Germany academic inbred. I have been at the same institutions so far. We'll never know what future will bring. We're doing good science here at Helmholtz Munich. We have good equipment, so I'm happy to be here. And today I'm happy to talk to you about information orthogonal to MS in metabolite identification. And this is sometimes the role of RT, CCS, and CO. So a short disclaimer, the CO will be a bit short. I will focus on RT and CCS, and RT retention time is something that's very close to my heart. So I see a lot of developments always on the mass spec side, and they are beautiful, they are important, but the other things partially get neglected. So this is where I would like to kick in and start changing that a bit. So what do we focus on? Very brief take from my side on metabolite identification. The use of retention time for metabolite identification, the use of CCS for metabolite and lipid identification. Very briefly, other things for metabolite identification, it's a small joke from my side and a short conclusion in here. And with that, I would directly jump into metabolite identification. And by training, actually, I'm a chemist. So what I typically start my talk with is the chemistry of the ohms and why metabolomics is complicated. So also here, short disclaimer for all the people. I don't know if there are any genomics, transcriptomics, proteomics people here in the audience. I will accelerate a bit, but it's just to give you, to set us the same ground for this talk. As I said, I'm a chemist, so I'm always coming back to the chemistry side of things. If we look into the chemistry of genome and transcriptome, so DNA and RNA, for a chemist, I would say that's pretty boring. These are linear polymers of only 4 building blocks. Okay, there's a bit of epigenetics in there. I'm happily ignoring that for the moment. But we have this linear polymer. We have approximately equal amount of all the bases, so we have a very limited compositional space of the things. And the most important thing that I'm always saying here is we have one physicochemical principle that underlies most of the techniques for genome and transcriptome analysis, and this is the complementarity of the strands. So you're always looking for hybridization, you always look for the complementarity in the strands. With PCR reaction, you can amplify, and then you always look into the sequence things that you have this complementarity in there. So we have the hydrogen bondings, and this is the physicochemical principle behind all these techniques. Of course, there are exceptions. There's nanopore sequencing, for example, for looking for the size, but generally, this is one common theme that could be used to analyze the genome transcriptome. Once it comes to the proteome, from a chemistry point of view, I'm still saying this is still a linear polymer. So I'm not talking about peptides, ribosomal peptides and so on. It's okay, really about proteins. Linear polymers with the amide groups. Okay, we have now 20 building blocks with different properties. But once you go into the analysis of proteins, what we do nowadays is we degrade them down to peptides. We analyze the peptides, and based on the fragmentation pattern, we infer the sequence, and with that, we can actually map back to the protein of interest. Again, for a chemist like me, boring. We're only dealing with peptides. But once we're coming to the metabolome lipidome, we have a lot of different structures in here. It's really, you see it here, we have very polar structures like trimethylamine N oxide, we have tryptophan, citric acid, we have cholesterol, we have here acetyl-CoA. So we have a lot of different molecules with a lot of different structures. And you already see it, we have rings, we have 5-membered rings, we have 6-membered rings, we have single bonds, we have double bonds, we have heteroatoms in the rings, we have rings without heteroatoms. And you can find one unifying principle of all these molecules in here. They contain carbon. But that is actually pretty boring for an analytical point of view of things. So this is where the complex task of metabolomics actually start. We have this structural diversity. On top, we have a large concentration range. So the example I brought you today here, these are the top 100 metabolites in a stationary phase E. coli. So you can see actually we have here glutamate as the top one, and we have adenosine here as the bottom one. These are the top 100. And if you look here on the scale of the y-axis, actually you can see here E+1, E-3. So we actually spend several orders of magnitude only in the top 100 metabolites. Of course, this concentration gradient is also true for proteins, but the task is different. So whereas genomics, transcriptomics, and proteomics, they have always the sequence where they can map back, we don't have such a thing. These metabolites are not genetically encoded. The enzymes producing them are, but not the metabolites themselves. And this is the hard task we're having for metabolite identification. We don't have anything where we can map back to say, this is true or this is false. And this is why we actually need a lot of efforts to identify the metabolites. And as Ollie stated correctly in one of his talks, I don't know when I heard it, Oliver, but he had a very nice sentence in there. He said, without metabolite identification, we only do featureomics. So yes, of course, we can do statistics. We can say we have 100 different thingies detected and they are statistically significant. Without the metabolite identification, we can't do biology. So metabolite identification actually has to be very close to every metabolomic scientist's heart. Brief disclaimer, I will talk only about mass spectrometry. I'm not an NMR guy. I know NMR, but I have not a lot of idea about NMR. So here on top, you see actually some metabolites we typically measure. We have leucine, isoleucine, and creatine. And this is pretty obvious. This has been published by Kind and Fien already. So you have high resolution mass spectrometers. They're not always helping, but sometimes they are. You need an isotopic pattern to get these things in there. So mass spec actually helps a lot. And if you have a mass spec of a decent resolution, where we say here is about 10,000, yes, we can have good separation from creatine and leucine, isoleucine. But you can see here, these two are not separated. Of course they are not, because these are isomers having the same mass. So mass resolution is good. It helps. And if you can have high resolution, I would definitely say use this high resolution in there, but it only helps with isobars, sorry. But what about isomers? Well, you can of course do an MS-MS on them, maybe on your high resolution mass spec. You have a QTOF, you have a Q-orbitrap, for example. But especially very close isomers, structurally very close isomers, tend to have a very, very similar fragmentation. This is just one example from one of my students where she set up a method in here and did, of course, the standard analysis of leucine and isoleucine and said, okay, I cannot differentiate them by MS-MS. And in this case, this was correct, because we actually started measuring here at master charge ratio 70, and the one fragment that could start differentiating these things is at 69. So if you are unlucky and your settings or simply your structures are so close in their structural composition, you cannot differentiate them. So you will get a lot of hits. I can say, I don't know which isomer it is. And this is actually where all the things I will talk today about are kicking in the orthogonal information. Talking about metabolite identification, we always have to talk about the levels of confidence. How sure are you in your metabolite identification? I think you all know this table from Emma Szymanski's paper. So I actually prefer this one over the original MSI one, because it's a bit more detailed in here, especially for level 2. You have this differentiation in here. But the most important thing for me is, if you have a look, this is heavily mass spectrometry centered. Only on the highest level, something orthogonal starts to kick in, the retention time, and only because we are measuring now the reference standard and we compare against a reference standard. Of course, we have here some experimental data, kicking some experimental evidence, but really talking about retention times is up here. So I'm always a bit puzzled. How can we actually use retention time in a much earlier stage of metabolite identification? Typically, it's used once you did your MS-MS. You probably run against a library in-house or external. If you have it in-house, you have the retention time already. But if you run externally, you probably get a good hit. They say, oh, that's the standard I have to buy. You buy the standard, you run it, you match the retention time, you match the mass, and you match the MS2. But couldn't this be used at a much, much earlier stage already? And this is where we're going to take off now with the first chapter here, retention time for metabolite identification. So if we talk about this task and going back to the same metabolites that we already had here, leucine, isoleucine, and creatine, we can see we could separate them by mass. But now here, if we take some good chromatography, we can start to separate leucine from isoleucine. And these are examples of methods we have used in the laboratory in the past. So this is a Kinetics F5 column. It's doing a great job, and you see here actually we get very nice separation of leucine and isoleucine in positive and negative mode. And this is obvious because here we are not doing anything fancy about the gradient or so between positive and negative modes. Same chromatography, just different ionization modes. So that's fine. Also with Hillig methods, so for the Hillig C from ethylene in negative ionization mode, we get, okay, it's not completely baseline separated, but we get separation. And for the positive mode, also here, we get actually quite a good separation of the two. So there are possibilities to do that. The question is always, which chromatographic mode shall I use, reversed phase, helic, or both? I don't want to cover this too much. So there are many, many possibilities. We have a lot of different modes, and we have a lot of different methods. And that's the most important thing is, even though people tend to use the same columns, we have very different methods. And this is something that bothered me a bit, is that, is there any chance that we can leverage the information from a different laboratory? And yes, there is. Actually, there's a very nice solution coming from gas chromatography, which is called retention time indexing. So the idea is here that you're not reporting actually the absolute retention time in minutes, but more like a relative retention time where you normalize the retention of a substance to known other substances. And in GC, this would be, for example, N-alkanes, and you can convert the retention time to a retention index. But this you can normalize for differences, for example, in column length and other factors. So the question was always, is there something similar we can do in LCMS? And actually, it was a very happy accident on one of the HPLC conferences we had over here in Europe. So it was a poster session. Actually, the only thing I wanted was to have a drink. So I was heading towards the table where the drinks have been served. And I bumped into a guy called Michael Quilliam. He's from the National Research Council in Canada, working actually on shellfish toxins. And on his poster, he showed a very nice ladder of substances, homologous series. And he was actually presenting his first takes on retention time indexing for LCMS for the identification of the shellfish toxin. And this is where he got me hooked up. And we started actually to collaborate on this project. And he sent me over his substances he uses for retention time indexing. These are called N-alkylpyridinium sulfonates, or for short, NAPs. So when we're ever talking about NAPs, we mean these substances. So you can actually see here is the pyridinium ring. We have this sulfonate group here, and we have an alkyl chain of different lengths. So it's starting here from 1 carbon up to 20 carbons. You can have even longer ones if you want to, but these are the typical ones we are using. The nice thing about them is they have 2 permanent charges. So they are very easy to ionize in a mass spectrometer. They are actually virtually independent of the pH, so you can use them at any pH range for retention time indexing. And if you have a UV detector, you see, okay, we have here a chromophore, so they're also UV active. These properties actually make them a very interesting class of substances for retention time indexing. So what we started now was evaluating, and we published this in 2021, can we use this for normalizing retention time information in metabolomics? And in this case, we focused only on the reversed phase for the moment. So what you see here, this is this ladder. So we have very nice separation, and you can see really like a string of pearls, one after the other, eluding very, very similar to the N-alkanes we use in GCMS, for example. We tested different interpolation possibilities, so linear interpolation, spline, Akima splines, and so on. It turned out that actually the linear interpolation is the best solution at the moment, especially at the higher ones. Actually, there's not a lot of difference, so we use actually the linear interpolation to convert retention time to retention indices. And to check if this actually is working, we started off measuring a lot of different substances. So we use this library, which is available from Aroa via Sigma, now Merck. And we varied some of the analytical parameters a bit. So our standard method was actually running at a flow rate of 0.3 mils per minute. So we varied the flow rate. So we went to a lower flow rate like 0.2, but we also went to higher flow rates like 0.4. So what you see here actually on top is the plot of the reference retention time versus the retention time at a specific flow rate. And on the bottom, you actually see the histograms of the percent deviation from the original method. And as obvious, if you use a lower flow rate, you will have higher retention times. If you have a higher flow rate, you will have lower retention times. But the most important thing is we have here actually quite substantial variation. So it can go up to 50% of deviation. So it's here 50, 25, typically -25, -25 to -50% of deviation, which means, okay, retention time is not the same. Okay, we knew that. That was obvious. So when we now convert the things to retention indices, because the snaps will be affected in a similar fashion by the chromatographic flow rate as all the other substances, can we use this? And actually what we see is here now, everything is now perfectly on the half line, and actually the deviations, you can see everything is centered around 0% plus minus a few percent. So yes, we can use this to normalize fluctuations in there. So we actually used some small flow rate changes to see typical fluctuation in the performance of the HPLC pump. We did the same with temperature, and it's also working. We then ask, okay, what about different formats of the column? So we used an Ascentis Express column here from Sigma, since it was a collaboration with Sigma. And we had one in an UHPLC format where we did most of the measurements, and then we had also an HPLC format column, which is then different in the dimensions and of course particle size, but it's the same chemistry. What you can see very obviously, we have here the retention time on the UHPLC, we have the retention time on the HPLC. They are not matching, but once you do the conversion to the retention indices, we can actually see here a very, very nice match up of things. This is what's already known from GC, for example, so you can use this actually the conversion from retention time to retention indices. So yes, it works. This was done for standards so far. So we went into C. elegans, like Oliver already mentioned. I was working on C. elegans for quite some time, and I'm still doing it, mostly human studies now, but C. elegans is still one of my hobby projects on the side. So what we have done is we have done all the measurements from the UHPLC on our water security. We have done the HPLC measurements on an Edgeland 1200. So what we did now is we use now the HPLC column in the UPLC system to simulate a third system. So the retention times should be similar to the ones in the 1200, but not exactly the same, because we have different depth volumes, we have different gradient delay volumes, and so on. So we measured some C. elegans extracts and checked, can we use the database we generated before on the UHPLC, on the HPLC, to identify metabolites based on mass, retention time, and if we had also an MSMS. So MSMS was working, so we used here MONA, Mass Bank of North America, tryptophan. By the way, my favorite metabolite is matching. Riboflavin is matching MSMS-wise. Okay, not the best similarity score, but intermediate, but more important, the retention index is matching. But also here metabolites like N-acetylphenylalanine, which are very low here in C. elegans, too low to be triggered for an MSMS. We had a very nice match of the retention index. And what we see actually down here, so this table is what I really like, is if we take tryptophan as an example, so we have this system number 3. So this is the HPLC column in the UPLC, which we have not been used beforehand for checking if the conversion is working. So this is a completely new system. We have the mass detected, we have a certain retention time. If we do the conversion to the retention indice, we have this retention indice of 472. So if we're going to match now with the retention times on the UPLC, because we have the shorter columns, smaller inner diameter, different flow rate, we would have retention now of 2.71 minutes. Okay, that's not matching at all. So you see actually the deviation is 46%. If we go to the HPLC system, which was the 1200 from Edgeland with the same column, we see we have 5.29 minutes, so already a 5% deviation. Closer, but not close enough. So once we do the conversion to the retention indice, actually you see we have now a very good match. So we are here within 2 to 3 retention indice units, and we are actually below 1% of deviation. So this enabled us really to have the retention time information used directly for metabolite identification. The next question we asked, okay, it's working within one lab, so this was the BGC at that time, working on different LC systems. Does it also work between different laboratories? So we started off with a small ring trial, and in the meantime, the ring trial has grown. So this is the first results from the 99 substances that have been distributed to the laboratories. We used the same chromatographic method. Actually, in this case, it was a Kinetics column The nice thing is retention times did not match, but the conversion to the retention indices is working. So this works also between the different laboratories. So we have a good solution for using retention time information also early in metabolite identification, because you can use the retention indices from some other lab, for example. So this is currently under preparation. The results look very nice. We have some more. I don't want to go into detail here anymore, so I can tell you it's working. The only problem we have in here is we did the test. There are people out there that say C18 is C18, right? It's a C18 column. This is not true. We all know each C18 column has a very distinct chemistry. Of course, the main separation is the hydrophobicity, but there are also secondary interactions and also the strength of the hydrophobicity. What's the carbon load on the column influences things. So we did a test, and actually retention indices from one C18 column to another C18 column are not comparable. So we have now a good solution if people are running same column, same solvents, very similar chromatographic systems. And this could be something like you have the things like the National Phenome Center, you have multiples of them, and you need to normalize differences in hardware, for example. That works very nice. But once you go out into the wild, you can see we have a lot of different things going on. So this is from one review of my PhD students. So what we did in the upfront of the part I'm talking next, we checked what has been used out there. So we used Metabolomics Workbench, we looked into metabolites and checked which columns have been used. So first thing we see, mostly reversed phase studies. A little bit of other things. So these are more like Kinetics F5 and some cyano chemistry. We have here a bit of Hillic. The good thing is this is actually growing over time, so people are aware polar metabolite analysis is important. But here we have a lot of different columns coming in. So there is not a single method that is harmonized across the different laboratories. And this harmonization is very unlikely to happen in future. So we need things like retention time mapping or prediction. So retention time mapping, there have been several papers out there also on retention time prediction. And together with Sebastian Böcker from the University of Jena, this has been a topic we always wanted to work on, retention time prediction for metabolite identification. We got a project funded some years ago, and now we took the chance, let's do that. The first thing we realized is we're falling short of training data. So our high hope was actually that within the different repositories that we have, we would have plenty of training data we can use to develop some fancy machine learning algorithm to start retention time prediction. And actually, we were proven wrong. So I went to Eva, my PhD student, and said, here, look into repositories, get the data out so we can use it. And after a few weeks, she came back pretty disappointed that I looked into the data sets. The quality is, well, not the best. Not talking about the data quality, but the quality of data we need for retention time prediction in here. So what we need is actually, we need data from reference standards where people are sure this is this metabolite, exactly that retention time. And more importantly, again, for us chemists, or at least for me, it's obvious, retention time is not retention time. So column, what you see here, actually matters a lot. And this was actually where we were falling short, that the data was there, but the metadata was not good. So we could not actually trace it back to which column has been used, which solvent has been used, because this is something that we wanted to incorporate into our machine learning algorithm at that stage. One of my favorite examples I'm always citing in here is a data set from metabolites where basically their description of the LC-MS setup was, we used a C18 orbit trap, where I'm always saying, okay, there are more than one C18 column and there nowadays are more than one orbit trap. So you should be a bit more specific on that. And with that in mind, we actually started to develop our own repository for retention time. This is what I will talk now. RepoRT, which has been published very recently, and I see now actually I copied the old slides where it was still under review. So it is now published in Nature Methods in there. This is our repository for retention time data. This is our repository for training data for retention time prediction. It's all hosted on GitHub. Actually, we like GitHub a lot. So not only for the code and sharing of code, but you can share anything that's based around text files. And it's very easy to use in that regard and very useful because you can track back changes through the commit system we have here in the GitHub. The important thing is here, from the beginning, we started to incorporate really the metadata. We said, okay, the metadata really matters. We have to know which column it was, which solvents has been used for preparation of the eluents, which additives are in there, what's the flow rate, what's the temperature, and so on and so on. So directly from the beginning, by design, these things have been included into repo RT. So we can upload the data in there, so it's open for submission. We will see it in a moment. So we also elaborated here a bit on these things. So what do we have? Well, we have the infrastructure now. RepoRT is hosted on GitHub. We have the data. Actually, each study we had an individual folder, and we have in each individual folder, we have distinct TSV files where all the information is stored. One of the most important tasks for us here was actually to make RepoRT useful for other people. And with other people, I'm not talking about metabolomics scientists or chemists, no, for machine learning people. Machine learning people typically look for new challenges to develop new algorithms, to test the latest algorithm they have developed for a different thing. And they don't want to learn what is retention time, what is the input I'm using for this. They want to have clean data where they have a clean task to do. Say, okay, this is the variable you have to predict, and these are all the input variables we give to you. So we started to design RepoRT in exactly this way to have it. Sorry?
Oh, good, just go on.
Okay. So we designed actually RepoRT directly in a way that it's machine learning friendly. So you can download the data sets and use it directly for testing and developing your machine learning algorithms in there. So we have all these individual files. We have certain files. You have an input folder, which is the raw data. We do some processing on the run, and you have then a data, which is the process data folder, where you have all the outputs which have been cleaned. You get some reports on this. You get the clean metadata. You get the fingerprints. You get molecular descriptors calculated by CDK in there. So you can directly use these things and start working on your other algorithms. We have an automatic standardization of the data. For example, we're running against the standardization of PubChem, just to have the same representation of all the structures in there. We do some automatic QC, so we're flagging actually duplicate entries, potential doublets where we have the same metabolite at different retention times and so on. And you get a report on every data set, and we're continuously improving this over time. This normalization is actually very, very important for us to really have a same standardized representation, not to deal with the diversity. How can you actually draw a molecule, but to have one standardized thing? And the important thing for us also is stereochemistry. Stereochemistry matters a lot. So these are some bile acids where you see they all have different retention times. But if we look into the atom connectivity, they're all the same. And this is typically what we call the canonical smiles, and these are the isomeric smiles. So we also have the different sub-data sets where we have, if available, the isomeric smiles or the canonical smiles in there. This is important for us to also have this separation. So we are here on good track to keep these things separated. And then it's the question, how well can we actually predict these different things? What do we cover? So at the moment, as of the date of publication, we increased now a little bit. We cover close to 400 data sets, mostly RP, a bit of HILIC. We have close to 9,000 unique compounds, close to 19,000 retention time entries. So that's the pair of retention time metabolites in there. We have certain data sets which are very, very similar to each other in the frame of the shared compounds, for example. So we generate several own data sets. where we systematically varied, for example, flow rate or temperature, or we used a different column with the same solvents. We used same column with different solvents just to have a bit of variation. They are very similar in the coverage of the metabolites between the data sets, but we also have data sets which are very different from the others. And for good machine learning algorithm, or for good machine learning solution, you need both, actually. And the most important thing is we want your data. So you see here, Uncle Sam pointing at you. We're welcoming actually data submissions to RepoRT. So if you have retention time data based on chemical reference standards, we're happy to take it. And if you're not fancy on working with GitHub directly, we have recently developed a GUI, which actually helps you uploading everything to GitHub and runs the automatic checks for you in the background already. So these are just a few screenshots. So this is actually ready to be used. So we have here some uploads. So you can upload, for example, a TSV or an Excel file. Actually, we have mapping functionalities that it looks into the header of your data set and suggests, okay, you named it RT, all capital letters. We use RT, all lowercase letters. So there's some automatic mapping which you can review in there. There's a review of the data itself where you say, okay, look, there are certain metabolites duplicated in there, or we have actually for the same metabolite multiple retention time entries. Are you sure that this is the correct thing? And we also have here the metadata entry. We have a huge database of columns in the background. So we curated tons of columns from different chemical vendors with the lengths, inner diameters, particle sizes, just to have all of this normalized. So this is RepoRT. Of course, we are also using this in the background to develop our retention time prediction algorithm. We're close to be ready to present it. So unfortunately, we didn't make it in time for today's presentation. A short spoiler here. So what was really important for us for retention time prediction? A lot of retention time prediction models so far work in the way that you have measured 300 substances, and with these 300 substances, you can predict the 301st. But people said, you know, that's not very interesting. Actually, would like to measure only a very low amount of substances. So we wanted to aim for something that can go de novo, if possible. And we have now some machine learning algorithm solutions in there, some machine learning tools which actually can do that. So our current best model actually works with the same accuracy as all the other models that has been published so far. The only difference here is our algorithm doesn't need to know anything about your system up front. So you don't have to put in 200 metabolites to measure to predict the 201st. So we can directly go with a low number of metabolites. Okay, so far for retention time. Let's move to another player in the field. More and more people are buying ion mobility instruments, and with ion mobility, you can measure the collisional cross-section, which is a proxy for the size of the molecule. And there has been many things where they said, okay, this is one of the next big things for metabolite identification, also for lipid identification. I also got an ion mobility instrument some years ago, so I said, okay, let's give it a try. The question is, where does CCS actually fit into our levels of confidence? So at the time when Emma Szymanski released that, CCS has been around, but it's not been incorporated. So the question was really, where can we put in CCS values in our metabolite identification workflows? How good does it actually work? So we did part of our ring trial for the retention indexing on an ion mobility instrument, and some other people also use an ion mobility instrument. So let's say, okay, not only compare the retention time and index, let's also compare the different ion mobility instruments, and people have done it. So there's always some systematic shift in between. Here we are, for example, comparing our ion mobility system against the ion mobility system in Geneva, which is a Waters system. So there's a bit of a systematic offset, which you can see here, and there's typically the range which we see that these traveling wave ion mobility systems are always a bit off. The more important lecture for me was working with metabolites. These are the CCS values of metabolites that I used before. So creatine, leucine, and isoleucine. So actually we can see, okay, creatine we can separate quite well based on the CCS values from the others. Where you say, okay, Martin, you have done this by mass. Why don't you need CCS? And you're definitely right. You don't need CCS on top. The problem is it starts here where you say, okay, this would be now the interesting one, leucine, isoleucine. Where you see, okay, they are very close. And if we look into the difference, it's only 1.3% difference between the CCS values of the two of them. So actually, my take-home message on metabolites and CCS is actually, you have to be very lucky that if the isomers you would like to separate really have a difference in CCS values, because you need to be above a certain percentage to really get really good separation. And typically what we are using for matching CCS is about a 2% error. So you would see here, they are falling together into the same error range. So there won't be any difference or any use of the CCS value on top. There were some outliers. They are always there. We're investigating what went wrong in these ones. The more important thing is we tested it also with lipids. And actually, I see this is where iMobility actually helps a lot in lipid identification. In this case, we were interested in very specific lipids in C. elegans, which are only present in a so-called Dauer stage. So Dauer is German for enduring. I did not name it like this, but here, these worms here, this Dauer stage is actually resistant to desiccation, and it's a non-feeding stage that can survive for a long time. So people are actually interested, what's going on in there? What changes happen in the Dauer stages which enable this longevity in the stress resistance? And one thing that actually is present in this Dauer are called maradolipids. These maradolipids chemically are 6 and 6' deacyl trehaloses. So you can see the structure down here, and they have been described in some papers. So far, there is no reference data on the fragmentation available on any database. So we're actually wondering, okay, can we identify the same species that the people have identified in the previous papers? And can we get some additional information on additional structures? They used shotgun lipidomics, whereas we went for an LC drift time iMobility based approach. And we were lucky actually to get some reference standards from a good collaborator that synthesized them for us. So we used in this regard the Edgeland iMobility system, the 6560, which is a drift tube instrument, where you can see here the drift tube before the quad and the collision cell together with a 1290 Infinity 2. We used our standard lipidomics gradient we published already 10 years ago. Now, what we used is actually a data-dependent, data-independent acquisition for generating the data. And actually, what I can tell you is data-independent acquisition together with iMobility actually makes a lot of fun. And we use this for the identification of the maradolipids. So first, we checked based on the chemical standards and some Kendrick mass defects, can we identify potential candidates in here? And you can see actually we have the ones without a double bond, with a single double bond, and with 2 double bonds. Also within one homologous series, for example, with 1 double bond, with growing chain lengths, we can identify here that we also have a growth or an increase in the CCS values. So actually, this is very valuable because we can start now to dig out homologous series, not only on retention times, but on CCS values in here. More importantly, what we did is we combined the iMobility with data-independent acquisition. So we developed a semi-automated in-house workflow where we used iMobility cleaned extracted ion chromatograms for specific fragments in correlation analysis to identify if a certain fragment that was detected in DIA belongs to a specific molecule. And this is done here for standards, for example. So we see here the MS1 trace, and we have here the MS2 trace. This is a very specific fragment where you have the loss of a certain side chains. In here, we did this in negative ionization mode. We also get them some other fragments, for example, the trehalose minus water and minus 2 water, and also the fatty acid side chains in here. And actually the spectra we can reconstruct on this is working very nice. It turned out that here with 20 electron volts, we get very good readouts, and we can actually get all the information we wanted to have in here. So we use this now for searching new structures, and what we have learned from the fragmentation is that these masses 323 and 305 are indicative of potential maradolipids, because this is the trehalose backbone, so to say. So we have a region here, actually, where we see both of the fragments, and they are co-eluting here. So we said, okay, these are maradolipids. And I don't want to go in detail now here. On top of the maradolipids that have been described, based on this DIA workflow, with DIA mobility, we could identify several more. But we were interested in, what's that? This is exactly the same mass. In here, we have a lot of peaks in here. And this actually points to a class of lipids called lysomaradolipids, which also have been shown only once. So this is the lyso version of them, so containing only one fatty acyl chain attached to the trehalose. And there are no chemical reference standards available. So we can only go back to spectral interpretation and, and chemistry and orthogonal information. And in this case, it was retention time and CCS. So we use the retention time and the CCS values as an additional filter here for these candidates we have, because we know we're using a reverse phase separation. We should have some kind of a homologous series in here. This is what you see here. This is for the fully saturated ones, and this is for the monounsaturated ones along the retention time. But also here, this is now along the mass to charge versus CCS axis. So you see, we have very nice trends. We also interestingly have here two separate isomers for what is called lysomaradolipid 17-0. So we don't have an SN1, SN2 specificity here for this class, because the two positions 6 and 6' are equal in that regard. So we only typically have one isomer. So this might be isomers here based on the different fatty acid side chains that's bound. And since we have a good chromatographic separation, we could also get a very detailed and accurate calculation of the CCS values for them. And actually what it pinpoints toward at the moment is that we have on one molecule that might be the saturated, not the saturated, sorry, the straight chain 17 fatty acid, whereas the other one might be an iso branch chain 17-0 fatty acid. The important thing here is really that a CCS value is a very nice orthogonal information for lipid identification, because we have this homologous series in here. CCS compared to retention times are actually much easier to predict. So there have been several algorithms in there. The problem here is that for small molecules, for example, the information content is okay, you can use it, but it's not like, what I've learned is actually CCS value, if you use the iMobility, it's not like CCS value, it's telling it it's this metabolite, it tells you rather it's not this metabolite. So don't believe that from iMobility you will get the one ultimate answer, but you can actually rule out several possibilities that are actually false positive annotations in here. We expanded that now a little bit into some other lipid classes. So another weird lipid class in C. elegans are these monomethyl phosphatidylethanol glyceramides. So you see here glucosylceramides. We have here this ceramide, which is glucose moiety here on top, and we have here this very unusual monomethyl phosphatidylethanol. Also, no reference standards available. Fragmentation has been described by Boland et al. In 2017. So we looked actually within Bruker-Timstorff data we had based on MassQL, so mass query language, if we can identify structures or potential candidates for these molecules. And we could actually find the 3 core structures that have been described by Boland, and we could see a few more. And we could again use the CCS value here as a potential filter. So we said, okay, this is not only a hit by chance, but it really seems to be part of the series, because if you look here, we have a very nice line which connects all of these lipids. In the meantime, we also found several more of them. So actually here, the CCS value, and sorry for the typo, it should be CCS and not CSS here, gives you nice information for the identification of new lipid classes. The question I typically get, is this always important? Actually, yes, it is. So we measured over different life stages. So we could actually see that this is one example. It's a 22 carbon version, actually decreases in the first life stages and then very strongly increases over the different life stages up to young adults. This is something we're also further investigating. What does this mean now for biology? But that's again where I'm coming back, why metabolite identification is so important. Again, citing Oliver, without it would be featureomics. So we would see the trend along this metabolite, but without a structure, what would be the biological meaning? So you need to go deeper into these things. And there's one very last thing, and this is just from my side, some, I won't say stupid joke, but just something to think about. Other things for metabolite identification. What other tools do we actually have for metabolite identification? And I would like to point to one very, very important tool, and that's the brain. Think about the things. I have seen so many papers which where metabolite identification was carried out in a, I don't even know how to phrase it, in a very sloppy, a very, very bad way, where people have identified cocaine in plants and many other things, where I said, okay, this is not true, right? Okay, maybe the people working in the greenhouse have taken cocaine and it just ended up on the leaf. You never know. But please, use your brain. It's very important. Is this a very reasonable metabolite identification or not? The best mass spec, the best eye mobility instrument, the best retention time prediction tool actually is nothing in comparison to your brain. Please use it and not blindly follow the results that the different software tools give to you. With that, a very short oral conclusion. So information on target to mass spectrometry is important. I think we all know that. There are novices entering the field, and I have discussions quite a lot. People asking me, Michael, which mass spec shall I buy? And I say, okay, it's not only about the mass spec, it's also about the things up front you have. So chromatography, please think about these things in here. So I hope I could give you a glimpse. Retention time, CCS, is important. They are not the ultimate answers. So it's not like retention time alone will save the world. It's not about CCS will save alone will save the world. It's a combination of things. So please, in future, take into the equation not only the mass spec, all the other things. Take into the equation also your brain. Please use it for metabolite identification. I think retention time indexing is a very good and promising way to start standardizing retention time information within its limitations, of course. Retention time prediction, well, we keep on saying for the last year, Sebastian and I, that's the next big thing. And we're working really to make it a next big thing. And I hope with RepoRT, we now made the first step in the right direction. And now people can jump on that. And this is actually the reason why we made it publicly available. We could kept now all our training data for us. Said, no, there are people that are much smarter than us. So please, here is the data, use it and produce the next tool that will change the metabolomics field. And with that, I would like to acknowledge a few people. So first, for all the retention time work, actually, Eva Maria, she's about to finish her thesis, just a final correction. She did all the work on getting all the training data collected together. She measured thousands of metabolite standards in the different conditions and really handpicked a lot of these things because we really wanted to go for high quality data we want to put in our database. So if somebody knows how to pick a chromatographic peak, it's Eva now. And of course, Fleming, PhD student on Sebastian's side. So he did all the back-end work on RepoRT. All the people that have been involved, Sebastian, of course, for the kind collaboration. It's always fun to work with Sebastian. In parallel, we had funding also from the ANR DFG on working some network thingies, which in the last minute I decided to not include because otherwise the talk might be too long. I would like to thank to my new group, the MPC, the Metabolomics and Proteomics Core. So we have been founded in 2021. There has been a metabolomics core, there has a proteomics core, and we have been joined together. And ever since then, we didn't have the chance to take a nice group picture. There was Corona, we had a cyber attack on campus, so there was always a good reason to not come together to take a group picture. I hope we can change this in the near future. And with that, I would like to thank you, and I'm happy to take any question you might have.
Well, thanks. I give the word to Apana.
Wonderful. Thank you for a great talk. We have lots of questions. So let's just get right into it. You started out the talk for with regards to retention time for metabolomics ID, and you had introduced a table where you showed separation of leucine and isoleucine using two different columns. So the first question is, why do you use Hillick Z method instead of the Waters BH amide column?
Let's phrase it the other way. Why not? Right. So I told that C18 column is not C18 column, right? And it's much worse with Hillick columns. So they're very, very different in their chemistry point of view. So we tested a lot of different columns actually for Hillick, and Hillick is still. I don't remember what was the Star Trek. Oliver, you can help me. Das Unentdeckte Land, one of the old Star Trek movies, the undiscovered or the final frontier. So there's a lot of things we still need to invest into in Hillick and testing the different columns. In our hands, and this is just a personal flawed opinion, the Hillick Z worked the best. The Twitter ionic column had the broadest coverage of metabolites with satisfactory peak shape, because that's one thing at Hillick, peak shape is not always the best. So for us, Hillick Z simply worked the best, and this was the reason why we used it. I also used the amide, and it's also doing the job for amino acids. So nothing wrong about using the amide. This was just the Hillick column we used, and it worked.
Okay. Thank you for that. And on that same note, you had also shown that there are two different gradients for positive EIC Hillick versus negative mode. Could you explain why the gradients were different for the two different modes?
Yeah, so I didn't go into detail, but what we have seen, again, because of the Hillick chemistry and the Hillick separation mechanism, I'm actually a big fan of for untarget of generic methods, because with untarget you never know what to optimize for. You don't know the isomers you would like to separate, so you want to have a good generic separation, which works quite well for reverse phase. So there when we developed our new platform on a Cenotoph, we just said, let's use the C18 with a water C2 gradient. For Hillick, that's not the case. So positive, negative, we have very different substances behaving different on the chromatographic columns. So far, we didn't come up with a good and satisfactory solution that will work in positive and negative mode the same way. So what we're doing now is, for the negative ionization mode, we use a pH 9 buffer system, whereas for the positive mode, we use a pH 2 buffer system, just to get the best possible results from both ionization modes. And the negative mode, of course, will then focus more on organic acids, sugar phosphates, phosphorylated stuff, whereas the positive mode focus more on amino acids, amines, and all the other things that ionize very well in positive mode.
Thank you for the clarification on that. Switching over to retention time indices, you showed that NAPS compounds are used for indexing the retention time. Are these compounds commercially available?
Yes, they are. So they are available from the National Research Council in Canada. The only problem is you don't need to search for NAPS. You need to search for, it has an official name itself, RMRILC. So if somebody wants to know it, please drop me an email. The good news is I'm in contact with the Sigma people, and actually they will commercialize this potentially mid of 2024, maybe Q3 2024. Because they have seen great interest in these substances, and it's much easier to order these things from Sigma Merck than from the National Research Council in Canada. So in a short time, they will be available via Sigma Merck. We are ordering them versus via the National Research Council of Canada. Also, no problem. They have several thousand ampules of this frozen somewhere.
Wonderful. And there was a question maybe more about like the theory behind, well, not the theory so much as the methodology. How is NAPS RTI different from internal standard normalization?
Well, the thing is, you can also do the internal standard normalization. Nothing wrong about that. The problem about the normalization here is we wanted to have something more universal in that regard for the NAPS. So different substance classes are affected differently by small changes in pH or organic content. So there's nothing universal in here. And also the NAPS will be affected in a different way. But the nice thing is we have something that could be used in a more linear fashion for the interpolations. As I said, we use the linear interpolation. So you also can do the internal standard normalization. So there has been also papers where Oliver has been involved in gradient break projection of these things. So there are many different solutions, and they all have the equal rights to be there and to be used. So for us, the NAPS simply worked, and ever since then we are using them. So we are very practical people. If something works, we don't question it; we just use it.
It works, right? And that is always a good way to go. I think the next question is kind of along the similar lines, and they were saying that often internal standards are selected based on like similar chemical structures. That's what's considered when you're using internal standards or selecting them. Is NAPS also specific to a certain type of metabolites when you're fixing the retention time index, or did you mention that they were pretty general or that they just worked?
Yeah,
maybe
they work. Of course,
how you select
there are
them.
differences, and this is what we have seen, for example, with different substance classes. So if you look more detailed into the substance classes, you see that certain classes are more on the higher side of the error, some on the lower side of the error, even if you use the normalized retention indices in here. This is just by the nature that the small changes in pH affect metabolites differently, depending on the structure. It worked good enough. Where it's getting interesting is actually when we started comparing across different columns. So where we see actually, for example, that the NAPS often are not too much affected by the column choice. So other substance classes are completely shifting around. So I didn't have it in this talk, but for example, we did a comparison on different waters columns, and we had the BHC18 and we have the CSH-C18, which has this positive charge on the surface on top. We had sphingosine in there, and actually this positive charge is some kind of a repulsion in there, because sphingosine in our separation conditions is positively charged, so it's eluting much earlier on the CSH compared to the BHC18. Whereas the NAPS, okay, they're also a bit affected, but it's not a dramatic effect, whereas there were minutes in between. And we see other substances which are not really affected, and they have virtually this similar retention index across different columns. So talking about different columns, talking about the same column, there are always substances that are working better than the others. Again, we are very practical. We need practical solutions. So far, it simply worked, and all within a satisfactory error.
Great, that's good to hear. How many compounds are usually in the kit, I guess, that might be available for purchase later on this year?
It's a single vial. You get a single glass ampule of a water methanol mixture where you have this 20 compounds in there. So it's starting with C1, so only a methyl group, until C20. And this is typically enough to cover the standard reverse phase range, which you have if you do a water acetonitrile gradient. So the C20 is typically at the end of the gradient. C1 to C3 are in the void volume. Mostly it depends a bit on the chromatographic conditions. Sometimes C3 is already a bit out. So we say, okay, we just use everything above C3 actually, because everything else is just void volume, and we are not interested in that. But it's a single ampule of 20 of these 20 substances. We dilute them depending on the mass spec. We are using 1 to 50 or 1 to 100 or even more. So I think you get 1.2 milliliters of this. So basically 1 ampule lasts forever.
Great, that's wonderful. Let's switch gears to RepoRT. I'm glad you spoke about how important metadata is. We always run into that kind of same problem that, you know, once you get into interpretation or just looking into the data, there's always a lack of metadata. And it's so crucial to know almost everything about your study, especially when you're, you know, pulling off data off of repositories. So we do have a few questions on RepoRT. How do you make sure the retention times in RepoRT are correct? How many of them are from chemical standards, and how many of them are MSI level 2 annotations?
So we demand, if somebody wants to upload data to us, that it's coming from chemical reference standards. So we really say you should use chemical reference standards. Of course, there's no way to prove it. So otherwise we'd go to the laboratory and say, okay, show us the reference standard, show us the measurement. So far we believe that people, they say, okay, this is level 1 identification, or this is a chemical reference standard. So that's the real prerequisite. We don't take any lower things. We had the idea of including them. We dropped it for the moment. What we're doing to ensure the quality, and that's always a question, is the retention time right? We had a lot of discussions about it. So we have a very, very simple retention time prediction model in there for quality control reasons. And this retention time model should not accurately predict the exact retention time for the system. It just should show us, okay, it's likely that this is the correct retention time, or it's very unlikely. So we use actually some interquartile range of the prediction of the retention times for one specific data set. And everything that's out of, I think, 2 or 3 times the interquartile range, I need to check in detail, this is the work of Fleming, will be flagged and say, okay, we believe this is very unlikely to be the retention time of this substance. Please double check. And if people don't double check, we simply throw it out because we are not really sure that this is the good one. So we have a very simple retention time prediction model in there based on gradient boosting to check how plausible it is. Not accurately predicting it, but at least giving us an idea, okay, this might be a plausible retention time.
Great, wonderful. You had said that diversity is key when you were talking about RepoRT. How do you ensure or benchmark the chemical diversity in the repository?
So we have some protections. So this is work of Fleming Kretschmer. He did in parallel to RepoRT. So we look actually into some scores about chemical similarity, where we can map out the chemical space on a UMAP actually, and this is one of the supplementary figures I didn't show. So we actually see that the chemical space is covered quite well. We have some blind spots. Every repository has that. So same is true for mass spec. So what do we have as mass spectral libraries? You typically buy the usual suspects, right? And then you measure them, and then you upload. So here it's the same. So we try to, so we are completely agnostic. Actually, we're not talking about metabolites, we talk about small molecules. If you have pesticides or something else, we have been contacted by people that have extractable and leachables in there. So drop it there. It's not about the biological meaning, but it's about the chemical structure we would like to have. We want to have a diversity in structure. So whatever it's a small molecule, just put it in there. We have some things running in the background for our own data set. We do Tanimoto similarities, maximum common substructures, and so on for our models to ensure we have good enough diversity. And at the moment, we're covering the chemical space, I won't say quite well, we cover it well. We could have a few more metabolites in specific areas of the chemical space, but this is something we're working on. This is actually part of the second project, is to identify what kind of structures we are missing, you buy the any chemical and in reasonably cheap and good amounts. And here we also don't care if it's a chemical, if it's a metabolite. We just say, okay, if it is a chemical standard which has a structural feature that would be important for us because we're missing it, we buy it. The interesting thing is Sebastian did this already. We're coming up with actually some very cheap chemicals we can buy, but the problem is you probably have to buy a ton of them. So you get actually a full wagon of these things for a few euros because it's some building block for chemical synthesis. But we said, okay, we need a few milligrams and that's it. But apart from that, I think we are doing well. It won't be perfect, but we're getting better.
Great, thank you for that. Switching gears to CCS for metabolomics and lipid ID, or metabolite and lipid ID. How do you find the SYNAPT G2SI in terms of using for metabolite CCS application? Is it sufficient enough? Also, did you encounter or hear some problems with this instrument with lock mass correction when using ion mobility?
I never had my hands on a SYNAPT G2. At the very beginning, when I started my PhD, we had a SYNAPT G1, where also Waters started to admit that this instrument could have been better. Was not the best instrument they ever built. I never used the SYNAPT G2 on my own. What I see actually is that it's not only from our data, but also from other data, that this traveling wave instruments are always a few % off compared to drift tube. Actually, drift tube is the reference method. So there is this depth field reference method, which is the method to determine a collisional cross-section. And the trapped ion mobility is also in the range of the drift tube. So these traveling wave are always a bit off, which is it's not horribly bad. It's not like you have 200 angstrom squared in the one instrument and 100 angstrom squared. So it's really, we're talking about a few % in here. The instruments within themselves are very reproducible. There have been also several papers showing, for example, Ian Wilson has been involved in these comparing different Waters instruments. They are reproducible within the different brands. It's just what we have seen that between the different types or the different types of ion mobility, there is always a bit of this systematic offset. And this is, you have to be aware if you want to match drift tube versus drift tube or drift tube versus traveling wave ion mobility. And if you know that, and if you can live with that, again, use your brain and then it works. And with the log mass, I have no idea because, yeah. Sorry, yeah, please.
Yeah. No, I was going to say, I appreciate the use the brain method.
Yeah.
There's one last question here on the slide where you showed CCS for identification of meratolipids, something new that I've learned today. How big of a measurement variation, RSD, do you find in this application when you use CCS? And how big of a threshold did you use for CCS matching? I think you had mentioned a percentage that you use.
Yeah, we typically use 2% and below as matching, and actually the measurement themselves are very reproducible. So we're talking about less than 1% RSD in these things. Depends, of course, how well you tune and how well you calibrate your instrument from day to day. But we are typically, if you do it very well, way below 1% for our drift tube. We also now have a timsTOF in the laboratory. Also there, we are very reproducible if you tune it and calibrate it in the right way. That's the most important thing. But we are low, be 1% for the reproducibility intraday.
Okay. There's one question in the chat remaining, and it goes back to the Helix Z columns that you had mentioned that you used. And it appears that you have used many columns, so it's good knowledge to have, and in terms of experience. Have you tried Helix Z that is peak line that provides good peak shape for more polar compounds like NXPs and NTPs?
Okay.
So it's a specific column.
Yeah, actually, yeah.
Yes.
Yeah. I didn't mention it in this slide because it wasn't important, but this was all the peak-lined hardware. So the important thing is metabolites, and particularly nasty metabolites like ATP, ADP, AMP, basically everything that has a phosphate group somehow likes to chelate iron. So wherever you have stainless steel in your flow paths, you still have iron in there. And this is why you sometimes have very bad peak shapes for phosphorylated compounds, also for carboxylated and polycarboxylated compounds, because you have this chelating effect. One big surface where you have a lot of stainless steel is typically the inner side of the column. And this is why Agilent produced this peak-lined version. Peak itself is not pressure stable, but if you put a metal column around, it's stable enough. So it's basically a steel column where you have a peak column inside, and there is the helic material inside. And with that, you are reducing this chelating effect, and by this having peak broadening in there. What we have seen, we recently bought a second Cenotoph, and we bought an Agilent 1290 Bio-LC with it, which is actually normally not used in metabolomics, but this instrument will be very specific for our helic analysis. And this is an iron-free flow path. So there is no stainless steel inside, but there is this alloy MP35N inside. And we have seen much better peak shapes for specific metabolites because there's no more iron in there. And this is exactly the same effect. Waters has something similar. They call it the not max quant, that's proteomics, max, something with max in there, where they also have some surface covering technology, which reduces the exposure of iron towards the liquid side of things. And with that also the unspecific absorption and the interaction of these phosphorylated and polyphosphorylated substances.
Great, thank you for the clarification. One more question came up in the chat. Do you need to do passivation to the LC regularly to achieve the reproducible data for the Helix Z column?
On a stainless steel containing system, yes. On the Bio-LC so far, there was no need to do that. But we only have it since December. So go ask me again in a year from now, I can tell you. On our stainless steel system, we do this, we typically passivate with phosphoric acid.
Okay, great. Those are all the questions in the chat. We do have some time remaining if anybody feels like unmuting and asking a question, or if Oliver, you'd like to add more comments. But
no, thank you. I think it was a great lecture. Yeah, thank you so much, Aparna. Thank you, Michael, for the late time that you put up here from your private home. We very much value that. We learned a lot about C. elegans and whatnot, and naps, and let's take a nap. You got to take a nap now. And so thank you so much, and I hope to see you in Japan, Michael.
I will be there, yeah. Looking forward to meet many of you there. Come over. I think it will be a great program. So I'm a part of the board of the directors, so I have to do this advertisement block there. Still open for poster submission. Go there. And of course, Japan is always worth traveling, right?
That's fine. All right. Well, thank you everyone for joining, and see you soon for the next seminars.
See you. Thank you very much for having me. Bye-bye.